Hello, good morning. This is Ravi Shankar. Welcome to Botany tutorial online classes. Today I am going to talk about one more important cell organelle, lysosomes. In Greek language, lyso means digestion. Zoma means bodies. That's why these are also called digestive bodies. Digestive bodies. At first, these were reported and named as lysosomes or perinuclear bodies. By Christian Dewey. At first they were reported and named as lysosomes or perinuclear dense bodies by Christian Dewey. Lysosomes are small, round, vacuolar structures. Vacuolar structures having means surrounding by single unit membrane and having 0.2 microns to 0.5 microns in size, contain enzymes, hydrolytic enzymes, all hydrolytic enzymes such as lipases, proteases, nucleases, sulfatases, phosphatases, etc. And these are the digestive enzymes. Almost there are about 45 digestive enzymes are present. 45 enzymes are present. 45 enzymes are present. Because of these enzymes, these lysosomes are involved in intracellular digestion. Intracellular digestion. And these are present in animal cells. These are present in animal cells and in few of plant cells. Animal cells such as secretory cells, and pancreatic cells, leukocytes means white blood cells, spleen cells, spleen cells, and liver cells. In all these cells, in all these animal cells, these are present, and in plant cells such as meristematic cells, these lysosomes are present. These lysosomes are present. Right. And so based on the origin, that means the origin, they are developed from Golgi complex and also from smooth endoplasmic reticulum, SER. This is the biological origin of lysosomes. This is the biological origin of lysosomes. Right. Structure. Structure of this lysosomes. Based on morphology, content, and their content, functions, lysosomes are categorized by 4 kinds. Lysosomes are categorized by 4 kinds. Right. What are the 4 kinds? Let us look here. One is primary lysosomes. Second one will be secondary lysosomes. Third one is, this is second, third one is autophagosomes. And fourth one is tertiary lysosomes, or otherwise called residual bodies. These secondary lysosomes are otherwise called digestive vacuoles. So these are the 4 kinds of lysosomes based on morphology, content, and functions. Right, when you're talking about primary lysosomes are, these are small vacuolar structures. These are small vacuolar structures, also called, these are small vacuolar structures contain enzymes. These are also called storage granules or protolysosomes or virgin lysosomes. These are having the diameter of 100 nanometers. These are primary lysosomes they are small vacuolar structures contain enzymes, and they are also called storage granules, protolysosomes, virgin lysosomes, or the diameter of these lysosomes primary lysosomes are 100 nanometers. 100 nanometers. These are about primary lysosomes. Next, when you're talking this secondary lysosomes are otherwise called digestive vacuoles. Digestive vacuoles. These are comparatively with the primary. These are large-sized vacuolar structures. Large-sized vacuolar structures. Right. Large-sized vacuolar structures. These are how these secondary lysosomes are formed when pinosomes or phagosomes fused with primary lysosomes then the secondary lysosomes will be formed. Secondary lysosomes or otherwise called digestive vacuole will be formed. See, suppose this is the cell, nucleus, Golgi structures, and above this there are perinuclear bodies are present. These are called lysosomes. How this will be formed? When these lysosomes are large-sized vacuoles are fused with the primary lysosomes, then secondary lysosomes will be formed. The pinosomes are phagosomes of this structures are fused with primary lysosomes, then secondary or digestive vacuoles are formed. They contain enzymes. They contain both the enzymes. We need to keep remembrance. They contain both the enzymes. Both the enzymes means maybe catalytic enzymes and hydrolytic enzymes. Both the enzymes are present here. Catalytic enzymes and hydrolytic enzymes. Catalytic enzymes and hydrolytic enzymes. That's why, and the materials which are present in cytoplasm of the cell will be digested. That's why they are called digestive vacuoles. So this is about secondary lysosomes. Secondary lysosomes are otherwise called digestive vacuoles, and they are large-sized vacuolar structures. And how this will be formed means when they are fused with, when pinosomes or phagosomes are fused with the primary lysosomes, then digestive vacuoles are formed, right? And they contain both catalytic and hydrolytic enzymes, and because of these enzymes they will be involved in digesting of materials which are present in the cytoplasm. That's why, because of that activity, the secondary lysosomes, otherwise called heterophagy, that activity will be called heterophagy. Hetero means digestion of different. Hetero means different. Phagy means feeding. Digestion of different materials. That's why that activity will be called heterophagy also. Right? That's secondary lysosomes. Next, when we talk about the autophagosomes, Autophagosomes are autophagosomes Autophagosomes are, these are otherwise called cytolysosomes. These are otherwise called cytolysosomes. These contain enzymes. These contain enzymes. Right. These contain enzymes. These, because of these enzymes, they'll attack and engulf the cellular content of cells such as mitochondria or endoplasmic reticulum, etc. under starvation conditions, these autophagosomes can attack and engulf the other cellular contents such as mitochondria and endoplasmic reticulum and feed them. Because of this activity will be called, this particular activity will be called autophagy. Auto means self, phagy means feeding. Cell by itself can feed up the other cell content. Due to this activity, we can treat these lysosomes as suicidal bags of the cell. That is about phagosomes. Right? Understood, ma? Next. When we're talking about the last one, what is that last one? Structurally tertiary lysosomes. Or residual bodies. These lysosomes, for example, this is the cell. These are the lysosomes. These lysosomes, they collect the materials, undigested materials. They collect undigested materials from the cytoplasm of the cell by this lysosomes. Then these lysosomes are moving towards plasma membrane. They are moving towards a plasma membrane and discharge their content outside the cell by the process will be called exocytosis. This is otherwise called effachy. That means send the material outside, release the material outside of plasma membrane of the cell. So that means these will be act as excretory in function. So this activity is called as effachy. So therefore these tertiary lysosomes are otherwise called telosomes. Tertiary lysosomes are otherwise called telosomes because there will be end lysosomes. These end lysosomes will be automatically, they will be killed. they will be disappear. How they will be disappear? They'll collect the materials, collected the undigested materials from the cytoplasm, and they are moving towards the plasma membrane and bulge outside the plasma membrane and discharge their content into outer region of the cell. That activity will be called E-Phagy, and they will be discharge the material by a process which is called exocytosis. So these are the four kinds of lysosomes: primary lysosomes, secondary lysosomes, autophagosomes, and tertiary lysosomes, right? And what are the characteristic feature and function also we discussed just now. And now let us discuss the functions of lysosomes. Next, functions of lysosomes. What are the functions? First function is autophagy. Second function is heterophagy. Third function is suicidal bags. Fourth function is E-Phagy, or the different functions which are carried out by the lysosomes. Autophagy. Autophagy means these are autophagy means they'll feed the content on their own, and they feed the content and digest the content on their own within the cell. That activity will be called autophagy. And heterophagy means there are certain foreign bodies like viruses and bacteria. If they enter into the cell, these foreign bodies foreign bodies will be engulf and feed and attack in certain leukocytes. That activity will be called as a heterophagy. That means they feed other materials such as viruses and bacteria in certain leukocytes. That activity will be called heterophagy. And suicidal bags. Under starvation condition, they feed the content, they feed the own content like mitochondria, endoplasmic reticulum will be attacked and feed and digest them under starvation condition. That activity, because of that activity, these lysosomes are considered as suicidal bags of the cell. And E-Phagy, as I told you, these are otherwise called residual bodies. That means they may collect the undigested food particles which are present within the cytoplasm of the cell, and they move on to the plasma membrane and discharge the material outside. That activity will be called as a E-Phagy. So they will be act as a E-Phagosomes, suicidal bags, heterophagy and autophagy. So these are the functions of lysosomes. So with this we completed about lysosomes. So all the best. Have a nice day. God bless you.